If you look back at the flowchart I gave you in the last PowerPoint, you saw what happened to sugar when oxygen was present. Glycolysis breaks down glucose into two molecules of pyruvic acid. That pyruvic acid then enters the mitochondria, where it's further broken down, releasing carbon dioxide into the atmosphere. The Krebs cycle produces 1 ATP per pyruvic acid molecule, as well as attaching electrons onto the carriers NAD+ and FAD. Electrons are then delivered to the electron transport chain from glycolysis and the Krebs cycle. Energy from the electrons is used to pump hydrogen ions across the inner membrane. The difference in charge between the inner membrane and the outer membrane causes hydrogen ions to move through ATP synthase, attaching a phosphate group onto ADP, forming ATP. But oxygen is not always present. Some bacteria live in harsh conditions, like in our digestive system, where oxygen is not present, and sometimes oxygen is simply just not available. If you exercise vigorously, it's possible to deplete the supply of oxygen in your muscles. If oxygen is not present, the electron transport chain cannot happen. The electrons that come down the electron transport chain would not have anywhere to go once they reach the end, because usually they attach to oxygen, creating water. So the cell has a problem now. It doesn't have the oxygen to keep the electron transport chain going, and it's running out of NAD+, which is needed for glycolysis. In this case, the process of fermentation occurs. Looking at the diagram, it's obvious why having oxygen is an advantage. The cell can produce a lot more energy, breaking down the glucose molecule. However, glycolysis can happen very quickly, as long as the NAD+ needed is regenerated. Enter fermentation. When oxygen is present, the cell goes through aerobic respiration. In other words, the Krebs cycle and electron transport chain. If oxygen is not present, the cell goes through anaerobic respiration. In other words, fermentation. There's two types of fermentation, which type the cell goes through depends on the cell we're looking at. Alcoholic fermentation is the first kind of fermentation. If yeast needs to produce energy to do cellular work and oxygen is not present, pyruvic acid will undergo chemical reactions so that NAD+ is regenerated. In the process, the cell produces two waste products, alcohol and carbon dioxide. And yes, this is the same alcohol in beer and wine. In fact, this exact process is used to make beer and wine. Other kinds of cells, like our own cells, undergo lactic acid fermentation if oxygen is not available. During vigorous exercise, you may have experienced pain in a muscle you were using. This was caused by the buildup of lactic acid. But whether cells undergo lactic acid fermentation or alcohol fermentation, the purpose of fermentation is to recycle the NAD+ required for glycolysis. Besides just our own muscles, some types of microorganisms undergo lactic acid fermentation. After glycolysis, pyruvic acid is converted to lactic acid. Again, most importantly, the electrons from NADH are removed, regenerating the cell supply of NAD+, which is needed for glycolysis. Yogurt and pickles are also used making this process. Alcohol fermentation happens in yeast and some other microorganisms. Have you noticed all those holes in your bread? They're actually the product of alcohol fermentation. When making bread, yeast, warm water, and sugar are first combined. Once the yeast has begun eating a good meal, flour is added. The yeast get trapped in the flour and run out of oxygen, so it begins undergoing alcohol fermentation. Besides producing alcohol, alcohol fermentation also produces carbon dioxide. These bubbles are what causes bread to rise and gives bread all those little holes. In the production of alcoholic beverages like beer and wine, yeast are fed sugar and trapped inside a closed container. When the yeast runs out of oxygen in the container, it begins to undergo alcohol fermentation and produce alcohol as a waste product. After beer or wine gets to a certain alcohol content, the yeast die, essentially of alcohol poisoning. Hard liquor is made by basically boiling off the water to raise the alcohol content of the remaining liquid. So there you have it. Now you know how to make alcoholic beverages. Just don't try it at home. We'll try it in class, and you'll see how nasty the basic process is, and what a skilled brewmaster you need to be to produce real beer. But for real, science is all about experimenting, but we need to be safe about it, and you should rarely consume anything you make in the course of a science experiment.